Hey everybody. So this is our last video in our series of videos about the nervous system, and we're going to be talking about research methods. So what that includes is we're going to talk about the effects of brain damage and how that's been used in research, the effects of brain stimulation and how that's been used in research, and then we're going to talk about the recording of brain activity. Let's start with brain damage. The text highlights that there's a difference between ablation and lesion. Ablation is the actual removal of a brain area, and a lesion is actually causing damage to a part of the brain, but not necessarily surgically removing it. Sometimes surgeons will elect to do a lesion over an ablation because the lesion may be easier to do than the ablation. In research, animals have been used to see how the removal of certain brain areas or damage to certain brain areas can impact subsequent behavior. In humans, a lot of times what we've seen are case studies where someone happened to get injured, and so as a case study, somebody documented what happened to that person. And what we see with humans is that oftentimes the damage to a certain area of the brain is going to cause some kind of problem with their mind or their functioning or behavior. One example that the textbook gives is that Broca had a patient that lost their inability to speak after they had damage to a part of their left frontal cortex. And that became known later as, and is known now as Broca's area. The limitations that are associated with using brain damage to come to conclusions about the brain in general is that it's not controlled, especially with humans. We're not going to go around as researchers and purposely damage people's brains to see what happens. It's not controlled, it's not specific. So sometimes when we have a case study and we see this damage, we may not necessarily know what that might look like in somebody else. And again, correlation does not equal causation. So if we see the scan that, for instance, that a certain part of a brain is damaged, can we for sure conclude exactly what's going to happen with each person that might get that damage? So now we're going to move into the effects of brain stimulation. So we just talked about how brain damage can cause problems. So if brain damage can impair some behaviors, stimulation should increase it. That's a direct quote from our text there. In classic animal studies, they found that electrodes were not specific enough. In other words, electrodes are measuring electrical activity, and though we may be trying to capture with the electrical activity a certain area, the electrical activity that electrodes are actually measuring may go beyond what we're trying to measure. More recently, optogenetics have been developed. There were three steps developed by Deseroth, and we will go over those in just a moment. But basically, optogenetics is using light to control a more limited population of neurons. So in other words, becoming more specific. Whereas electrode it might be capturing more than we wanted to. The optogenetics is helping narrow down what we're actually capturing. So what they did is they developed this three-step process where they created a protein that would respond to light by producing electrical currents, and then developed viruses that could be inserted into one of these proteins and therefore into certain neurons. They then developed thin optical fibers that could shine light onto these neurons so that basically they could be more confident that they were capturing the areas that they thought they were capturing. So basically, people using optogenetics can be more confident by seeing this shine of light that they are capturing or measuring the activity in the neurons that they're trying to capture. So the last thing we're going to cover is the recording of brain activity. The recording of brain activity is a pretty big topic because there's a lot of different ways that are complex and there's a lot of information that goes along with these different ways that researchers record brain activity. So the simplest way is single cell recording. And that's when researchers use a single electrode to measure the activity of a single neuron. From there, we have the electroencephalograph, or EEG, and that is using multiple electrodes to measure brain activity. So oftentimes, the participant in an EEG study will be wearing a cap, and depending on the research or what the researcher is interested in, there could be 10 electrodes, and there could be 50 electrodes. So it really depends on the study, how many electrodes the researcher will use. A magnetoencephalograph, or MEG, measures faint magnetic fields. So it's similar to the EEG, but instead of that electrical activity, it's looking at magnetic fields. We then have positron emission tomography, or a PET scan or PET scan, and that's a high-resolution recording of emission of radioactivity from injected chemicals. So a participant using a PET scan would actually be injected with a radioactive isotope. We then have fMRI, which is a modified MRI, and that detects the amount of hemoglobin with oxygen. So this table I found is actually, let me move my camera over here so I can see that, so you guys can see that. So we've got the PET scan, and there's pros and cons to each of these ways of measuring brain activity. So the PET scan is less sensitive to motion artifacts, so it might have that higher resolution, but there's a lot of disadvantages. It's more invasive. Somebody's being injected with a radioactive isotope, low spatial resolution, low temporal resolution, and it's expensive. An EEG, a really awesome benefit of that is it's lower cost, and it has excellent temporal resolution. However, it has poor spatial resolution. An MEG, or MEG, is a high temporal resolution and good spatial resolution, but it's very expensive and limited resolution for deep structures. fMRI has good spatial resolution, is non-invasive and widely available, but it has a relatively low temporal resolution. The lab that I am involved in at UT Tyler is a psychophysiological lab, and we read a lot of articles, and one of the things we often see is that researchers may use an fMRI and an EEG so that they can get basically everything that they're wanting to. Because as you can see, one of the disadvantages of an fMRI is low temporal resolution, but that is an advantage of an EEG. So a lot of times researchers will combine those two so that they can get more information. So the last couple of things that I wanted to highlight in this video is that on page 98, there is a table that summarizes a lot of the things that we talked about in this video. And I also wanted to leave you with that correlation does not equal causation. So just because we have a scan that shows us that a certain brain area lit up for somebody does not mean that we know exactly what is going on or why it's going on. So thank you so much. This concludes our nervous system series of videos. And please, again, let us know if you have any questions. And I hope you have a great week. Thank you.